So, where were we going with this?
The iron.
Ferritin complexes with iron when iron levels are high. So, when iron levels are high, what's going to be the expression of this gene? Generally speaking. Sorry, so let's do low iron first. Low iron. In a low iron situation, ferritin isn't needed because you don't have to bind up iron, so you don't need it. And so what's going to happen to this gene in terms of its translation?
It's not going to be expressed.
Yeah, you're going to want downregulation. So what happens here is when you have messenger RNA for ferritin, you don't need ferritin when you have low iron. And so there is a protein, it's kind of like a transcription factor, only this is a protein that is involved in translation, and it binds to this iron response element, and it's called an iron response protein. If you haven't gathered this yet, scientists are not terribly creative at naming things. And as long as this protein is bound to this iron response element, you don't get translation. And so essentially you've transcribed the messenger RNA and you have gotten it ready for translation, but the presence of this protein bound there essentially prevents the ribosome from joining there and prevents translation from happening. When iron is in abundance, iron actually interacts with this iron response protein and strips it off of the iron response element. Now you have the IRP bound with iron. Because iron is high in abundance, that removes the translational suppression of this messenger RNA molecule. You start producing ferritin, and then these ferritin molecules bind with iron, and when they bind with iron, the iron is now protected from interacting with the cells. It's a way of essentially sequestering iron in most cases in the bloodstream so that it no longer is a toxic factor to the cells. So this element that this protein binds to is in this untranslated region of the messenger RNA. And so we kept drawing these untranslated regions in these messenger RNA molecules. We didn't really talk about what their function is, but one of their function is that these are where these regulating elements of messenger RNA are. Now, when we think about transferrin, when there's a lot of iron, we don't want transferrin there because we're going to get too much iron being transported into our cells. But when there's low iron, we're going to want to be producing this because we want to produce these transporters so we can transport iron into our cells which need it. And so in this case, what's going to happen is in the 3' untranslated region, there are 3 different iron response elements. All of these things have iron response proteins. And when these iron response proteins bind on this end, it stabilizes the messenger RNA molecule, keeps it from being broken down. So remember, what happens to messenger RNA molecules? They get transcribed MAD-cap style, and they get degraded really fast. One of the reasons that you have so much transcription of messenger RNA is because it's not very stable. It doesn't stick around very long. When you add these iron response proteins, it doesn't do anything to what's happening at this end where the ribosome is going to occur. It's going to add these iron response proteins here at the very end. That stabilizes the messenger RNA. So once a messenger RNA molecule gets formed, it sticks around for a longer period of time. And so then, if one bout of translation goes on this messenger RNA molecule, another one and another one and another one can happen on that same molecule because it's being stabilized. I don't know why they put the transferrin molecule here, but basically that's the molecule that gets built from this. If you have high levels of iron, you don't want translation of transferrin. And so these iron response proteins once again bind to iron. They are removed from the iron response elements. As soon as you get rid of those, this is now not stabilized. It tends to degrade at the same rate that other messenger RNA molecules degrade at. And as a result, you're going to get less production of this protein because you haven't changed transcription at all. You've just changed the lifespan of messenger RNA molecules. So you have this antagonistic response in two different genes, a gene that codes for ferritin and a gene that codes for transferrin, and they're both regulated at the translational level. But one of them has a regulatory unit that is on the 5' untranslated region, the other one has a regulatory unit on the 3'. It doesn't really matter whether you're regulating at one end or another. In this case, this is regulation that stabilizes the messenger RNA. At the other end, the regulation essentially prevents the formation of the ribosome coming together, the ribosomal subunits coming together. To basically regulate iron levels in organisms. Questions about that? In the screencast and in the textbook, there is all sorts of information about what types of RNA make up the large subunit, the small subunit, things like that. But I basically gave you a basic outline here. There's a much more detailed outline in the screencast. The next example in your book is about fruit flies. And so let's just go through this. This is not on the screencast, so this was where I hoped to start new information. When you look at Drosophila embryos, there is development that basically deals with the body orientation of the developing embryo. And so at one end of the egg, this is going to lead to the head end of the embryo. This end of the egg is going to lead to the tail end of the embryo. So there's this linear patterning that we've talked about in the past. And that's governed, once again, like we've talked about in the past, by lots of different genes acting to set up that body segmentation. But two of these genes that are involved in this are the hunchback gene and the nanos gene. And so whenever I see nanos, I think of Thanos for whatever. Anyway, this is, the shorthand is HB for hunchback and NOS for nanos. But these proteins, NOS protein is distributed in the egg such that there's more of the NOS protein at the posterior end and less of the NOS protein at the anterior end. Messenger RNA that is put into this egg by the female is spread uniformly throughout this egg. And so what happens, because hunchback messenger RNA is spread throughout, the way you get anterior to posterior patterning, one of the ways that you get anterior to posterior patterning, is that this NOS protein, which is at greater abundance at one end than at the other end, actually serves to regulate the translational activity of this hunchback messenger RNA. So what happens is, when you have a lot of hunchback messenger RNA in the anterior end of the developing ovum and developing embryo, you just have hunchback protein being made. And when you get to the posterior end, where there's a lot of this nanos protein, there's another protein called pumilio. That's what PUM stands for, pumilio. Pumilio, once again, interacts with a nanos response element, binds to that nanos response element. Once pumilio is there, NOS can bind to that. And once NOS binds to that, it basically shuts down the translation of the hunchback And so you achieve this gradient of translational expression from the front end of the ovum and developing embryo to the rear end of the developing embryo, simply because this protein essentially downregulates translation. As its abundance falls as you move anteriorly, you get less and less downregulation, such that at the anterior of the body, you're getting a lot of the hunchback protein being translated, and at the posterior end, you're not getting as much of it translated. And so, just another form of regulation in terms of how genes get expressed. Remember, this should be old hat to you now, DNA to messenger RNA to protein. We talked about transcriptional controls on messenger RNA production. We've talked about things that alter messenger RNAs and can regulate RNAs once they've been formed and before they're shipped out of the nucleus. And now we know that there are ways in which the messenger RNA itself can be involved in regulating protein production. So you don't just have to look at, and why did I spell proteins wrong? You don't just have to look at transcription as the only regulator. There are all these other regulators, and at the end of the class today, we will see, get to a general outline of what all of that is. Get in the habit of drawing things on Friday, assuming that we finish today on translation. Friday is going to be spent practicing transcription and translation. The test next week, guess what? It's about nothing but transcription and translation. That's all we've done since the last test. So we're going to have clay, we're going to have pipe cleaners, we're going to have all sorts of things, and your goal on Friday is basically going to be to build models of these things that are models that would be productive in you being able to walk yourself through these processes. While you're in your dorm room, sitting down and drawing these things is helpful. The craft supplies that I'm going to bring in on Friday, they sit in the hallway on the first floor right outside Dr. Ron Lee's office. They're just in drawers, they're not locked up or anything else. You can come by at any point and use those if that's one of the ways in helping you get this stuff down. So we'll be using whatever it is that you need to do on Friday to essentially review transcription, review translation in an effort to get this cemented in your head. And once again, always think of this as a process. And so what is the process of transcription? What is the process of translation? At what point does this get regulated, and what does it get regulated by in each of these things? So when you're building, if you're building a gene out of pipe cleaners, don't forget to add these cis-regulatory modules that are way upstream. You can use different colors of pipe cleaners and things like this to illustrate that. Yeah.
Something like how we did the labeling of the DNA replication kind of thing. You know how we labeled that and gave us like sheets to practice. Is there something like that for this? With the replication bubble? Yeah.
Oh, no, but I could probably put something like that together. Yeah. Would you like me to do that? Yeah. Sure. Also, pay attention to the animation. So pay attention to the transcription animation that I put up there. There's also a translation animation that I just didn't have time to load this morning, but I'll also put that on there. So Friday's practice, and we'll spend time practicing. Monday is your literature summary from hell, and then Wednesday is the exam. And then we'll be free of molecular genetics for a while. So your book has talked before about the code, the genetic code. I've talked about it before. This codon table, the codon table represents the order of RNA nucleotides and messenger RNA. It is not an anti-codon table, so don't get that confused. It tells you what all of the combinations give you, and this table just basically points out that there's a lot of what your book refers to as degeneracy, this redundancy in notice about all of this is a lot of that degeneracy actually is localized in the last nucleotide in a codon, and that's not surprising, really. We've talked about start codons. Start codon, the only start codon is adenine uracil guanine. We have these 3 stop codons. Since there's only one mutational difference between this stop codon and this stop codon, as it turns out, tryptophan is not really used very much. And so all of these proteins have different frequencies of occurrence in organisms. Lysine is super common. So when you look at lysine, it's a really common amino acid in cells, in proteins. And so you find that these things are relatively conserved, and they're conserved across all things. So if you look in bacteria, stop codons are the same, start codon is the same. You look in fungi, start and stop codons are the same. Look in all the animals, all the plants, they're all the same start and stop codons. That this is a universal code for the most part. These proteins have characteristics, and I've talked a lot, and I feel like it doesn't hurt to revisit this. I've talked a lot about polar molecules, and I was talking to Dr. Reynolds about this last week. And when you take a chemistry class, I think a lot of time is spent on covalent bonds, because those are the bonds that make up the substance, which is what people oftentimes study in chemistry. You know, what is the structure of a sugar? Well, a sugar is something that's held together by covalent bonds. But when you get to biology and cell molecular biology in particular, those bonds are too strong. Those are the bonds that hold things together, and they hold things together in kind of permanent ways, right? But what you need when you're having molecules interacting with one another is you need more transient bonds, bonds that don't stick around for very long and are easily formed and also easily broken. And so when we're looking for bonds that are easily formed and easily broken, hydrogen bonding is one of these things. How do hydrogen bonds form? Somebody remind me. Okay, so the electronegativity of an atom will pull electrons away from another atom, and oftentimes when we think about these atoms, we think about these atoms as being hydrogen atoms whose electron cloud is essentially pulled away from them towards whatever the electronegative atom is, in this case oxygen, such that the hydrogen has a partial positive charge, the oxygen has a partial negative charge. If this is a water molecule, this is the classic example. You have 2 partial positive hydrogens out here because their negative electron density is being pulled away from them. And if you have another water molecule nearby, the partial negative of the oxygen will be attracted to the partial positive of an adjacent hydrogen, and these things will form hydrogen bonds. This is part of the ability of the characteristic of water, and the water is cohesive. It tends to stick to itself. It's what allows surface tension to occur, things like this. So when we think about amino acids, individually, we can divide them up into whether they hold full charges, whether they hold full positive charges or full negative charges. But then we can also look at them on the basis of whether their side chains are polar or not. And so we have a whole section here in the middle in purple where these are side chains that have polar molecules associated with them. Once again, what do you see here? Oxygen. You see nitrogen attached to hydrogens, oxygen attached to hydrogens. What that means is that those oxygens and nitrogens are going to be pulling negative character away from hydrogen, making those hydrogens partially positive, and that makes this a polar side chain. The thing that doesn't change in here is the amine and the carboxyl group. Those stay the same. And when you add new amino acids, you're going to be adding an amino acid, adding the amino group to the carboxyl group, so that a final protein has an N-terminus and a C-terminus is how we usually refer to it. And then there are nonpolar side chains, and then there are some special side chains. This is a side chain that actually forms a ring structure, so this is a particularly rigid proline This is a side chain where the side chain is basically just a hydrogen atom, so it's a really small side chain. And this is the only side chain that contains sulfur in it. And so cysteine, which is what that is, tends to create sulfur-sulfur bonds with other sulfur molecules. And so in a protein, you'll get these cysteine bridges, these sulfide bridges that bond cysteine molecules, cysteine atoms together. And so you find cysteine bonding to itself a lot. So the reason that it's important, you don't have to memorize all of the amino acids, but you do need to know that some of them are positively charged fully, some of them are negatively charged fully, some of them are polar, and in all cases, the charges that are going to be on there are partial positives that are exposed. And then there are nonpolar molecules, and then there are occasional special molecules that can bond with one another. Because once you get this amino acid sequence, once you get the primary structure of a protein, the amino acid sequence, that thing starts folding up on itself to create more complex three-dimensional structures. And what is responsible for that structure holding together aren't covalent bonds. In the same way that the things that hold two strands of DNA together, those aren't being held together by covalent bonds. Those are being held together by hydrogen bonds. Same thing here. Lots of hydrogen bonding going on, maintaining the three-dimensional structure of complex proteins. I study the thermal biology of lizards. It's one of the things that I do. And lizards are ectotherms. They get their heat energy that warms them up from the environment. One of the challenges of that is that they don't create metabolic heat the way we do, so we're constantly regulating our temperature up and down slightly. They have to shuttle in and out of shady spots, sunny spots, moving around. They do what we call behavioral thermoregulation. Because that oftentimes their body temperatures can be more variable than ours is, it doesn't have to be. If you're a good thermoregulator in terms of behavioral thermoregulation, you can usually maintain your body temperature during a fairly narrow range of temperatures until the sun goes away. And then, once you don't have the ability to gain heat, you kind of go back to just being the same temperature as the environment. Well, as you heat up a lizard, and we do this, we cool lizards down and we heat them up, and we run them down racetracks to see how body temperature influences physiological capacity, we get what's called a thermal response curve, where temperature goes from about 40 degrees to about 15 degrees centigrade. This is pretty hot. Your body temperature is 37, and 15 is uncomfortably cold in the lab if you have to stand there for 4 hours racing geckos. But what happens is, as you heat a lizard up, there's a very slow ramp up of physiological activity, because in this case, a lizard is just a bag of chemicals, and so in the same way that you add heat to chemical reactions and they go faster, you add heat to the chemical reaction that is a racing lizard, and it goes faster. But then you get to this kind of sweet spot in the middle where there's this optimum temperature range where they're running fast. They are cooking. They're running, a little lizard like this will do more than 1.5 meters a second on a racetrack. They can be really, really fast. But what happens at this end? Does it start going down like this? No, how does it go down? It goes down like that. Why?
Proteins are denaturing.
So you say proteins are denaturing, unpack that. Somebody else unpack that for me. What does denaturing mean in the context of proteins? There's enough heat to break these transient hydrogen bonds such that it doesn't hold its three-dimensional structure anymore. The three-dimensional structure is gone, then the function is gone, and when your function is gone, you basically stop doing what you're supposed to be doing physiologically. So this precipitous decline in speed is because you're basically killing the animal, and you're killing the animal by denaturing the proteins, because the proteins are held together because of these properties of the amino acids. This is why we can do PCR, because we use heat to split apart the DNA molecule. It doesn't even have to be very hot, it has to be somewhat hot, but we apply enough heat to break those hydrogen bonds that allows new DNA to be made because of these transient source bonds. So I realize I'm now probably hammering this point home more point, but it's something that I saw in the last test that we just need to reinforce this. So, the thing that your book shows you, the thing that your book shows you is that oftentimes things that have particular character are all grouped together, they're clustered together, they're coded for by related codons. And so when we talk about something being hydrophobic, that's going to be one of these nonpolar amino acids that is just surrounded by a bunch of hydrogens. They don't have polar hydrogens on there, and so they don't interact with water very well because water is a polar molecule, as we saw over here. We have polar molecules that will form hydrogen bonds, and so they're called hydrophilic molecules. And then we have molecules that have full charges, and they'll interact with lots of things, but they'll interact in ways that form more permanent bonds because they have full charges, and so they do things like ionic and covalent bonding. But those are relatively few when you look at, in the grand scheme of things, 1, 2, 3, 4, 5 out of the 21 or whatever it is of the essential amino acids. When you look at changes in protein identity in a sequence, so this is a beta globin gene in a bunch of different mammals, bat, aardvark, human, rabbit, whale, mouse. One of the things that you'll notice is that there are some amino acids in the sequence that are invariant. They're the same across all of the different taxa that are in this table. And then there are a bunch of other ones where there's variation. And so you can see that in some cases they're pretty conserved and only differ in one particular taxon. There is variation from species to species in terms of what the final protein looks like because of things like mutation. But what we notice is that there are some areas that are highly conserved, some regions that are highly conserved, and then there are other regions that are somewhat less conserved. And also, you'll notice that certain kinds of proteins, or certain kinds of amino acids, take the place of similar kinds of amino acids in terms of their function. So oftentimes when there is a mutation, if you're replacing one polar amino acid with another polar amino acid, you can still retain function because the active site that that protein, that amino acid is involved in, still retains its functional character. And so there are things that are similar in charge. So these guys here, there's variation, there's not a ton of variation, but these guys are similar in charge and character. These guys are all similar to one another. These guys are all similar to one another. And so one of the things, one of the flexibilities of protein sequences, amino acid sequences, is that they don't have to be perfect, and they can still provide function as long as you're kind of maintaining the same type of amino acid in that particular place. So even when there are mutations that do change the amino acid sequence, that doesn't always create a non-functional amino acid. Read about this more in your book. This is a table that basically shows you what the substitution rates are. And so alanine, if you look at a bunch of different taxa for a particular gene, alanine at one site almost always means alanine at another site, but oftentimes alanine is replaced with glycine, oftentimes alanine is replaced with serine, oftentimes alanine is replaced with threonine. Alanine is never replaced with TRP, which I don't remember what TRP is. It's hardly ever replaced with tyrosine. It's hardly ever replaced with phenylalanine. It's hardly ever replaced with methionine. And so when you look at the same gene across lots of different organisms, you can begin to get a feel for what kinds of amino acids are interchangeable with other amino acids. One particular amino acid sequence that we talk about in evolution and ecology is when aspartic acid gets replaced by asparagine. And so aspartic acid gets replaced by asparagine, and that one change really changes the nature of a sodium channel in a snake, such that it takes a snake that was sensitive to tetrodotoxin and makes it 100 to 1,000 times more resistant to tetrodotoxin by just changing this one amino acid in this long sequence that makes up the sodium channel. But that actually turns out to be a pretty common mutation that occurs. And it's going from a polar molecule, it's going from a negatively charged aspartic acid to a polar amino acid, asparagine. And so it basically goes from a negative charge to a partial positive charge. And if you look at the structure of tetrodotoxin, tetrodotoxin has all of these hydroxyl groups hanging off of it. And hydroxyl groups are polar, partial negative oxygen, partial positive hydrogen. When those partial positive hydrogens interact with the partial positive hydrogen in asparagine, they repel one another, rather than when it's aspartic acid with a full negative charge, all those partial positives on tetrodotoxin bind, and they bind firmly to that. And so then you get blocking of this sodium channel just because of one small change in amino acid sequence. This is useful when you go to align protein sequences. If you're doing something like a blast search or a clustral search, knowing what the relative rates are of substitutions of amino acids and protein sequences is useful for computer algorithms if you're trying to compare amino acid sequences across lots of different organisms. That's originally why this was put together, was to aid in alignment. What did you guys do in lab yesterday? What did you guys do?
Just did bioinformatics stuff.
Just bioinformatics stuff? So have you done some alignments?
No.
No, not yet. You will before it's all said and done. Just know that this is the underlying basis for those alignments working. Questions so far? All right. Remember, histones get modified? Histones are just protein molecules. They have these histone tails that can get modified. Those modifications change the charge nature of those histone tails. That affects how histones interact with one another. You guys remember all of this? Okay. So a histone is a protein. The things that we're talking about here are proteins. And so all of the same kinds of modifications that you can do to histones, you can do to any other amino acid in any other protein. Remember what happens to a protein that gets ubiquinated? Yeah, it's a signal to the cell to degrade that protein. It's right down pressure. It's all that word. Ubiquination, that's a tag that gets added to basically signal that protein for being degraded. Remember that methylation and acetylation, it basically surrounds the amino acid with nonpolar hydrogens, essentially, so it makes it hydrophobic. If you add phosphorus, it takes it and converts whatever that amino acid is into a negatively charged amino acid when you get phosphorylation. That then enables it to interact with other things as well. And so you can get a number of different protein modifications. These protein modifications are added after translation occurs, and oftentimes they're really important in determining how these proteins fold up. The three-dimensional structure of proteins after they have folded becomes very important for their function. It enables them to interact with the substrate molecules that they're interacting with appropriately, and that becomes very important. Another thing that your book doesn't talk hardly any about, have you guys ever heard of chaperone proteins?
Yes.
What do chaperone proteins do? Yeah. Yeah, so these are other proteins that help other proteins fold up correctly. And we used to have a guy who worked here who actually was interested in chaperone proteins and worked on chaperone proteins and how they operated. If something happens to your chaperone protein that is responsible for correctly folding a particular protein, it not only affects the chaperone protein structure and function, but it also may make, even though you're transcribing and translating this protein, you don't get a functional product because something has happened to the structure of the chaperone protein. A lot of the heat shock proteins that we have, And a lot of the proteins that we know are linked to stress, things like oxidative stress and stuff like this, oftentimes these proteins are chaperone proteins. They actually don't have a function in the thing that is being looked at. They have a function in helping fold the things that have a role in that function. And so many of the stress-related responses are due to having these chaperone proteins. So you make this big, long amino acid sequence. Once it's made, it's not done because it has to be folded, but also sometimes it's not done because the final product doesn't have all of the different parts. And so when we talk about insulin, there's different forms of the insulin. When the insulin comes out of translation, you have this kind of, I think of it as a paperclip sort of shaped molecule. What are the amino acids here? Cysteine, it's still on the board. I didn't erase it, because I knew this was coming. So these are these cysteine linkages, these disulfide linkages that bind cysteines together. And in this case, they're keeping this pre-proinsulin molecule in its particular conformation. Not only that, you start with pre-proinsulin. The first thing that happens is this N-terminus section gets split off by proteolases, leaving you with, once again, still most of this paperclip-looking thing. But then, when you take proinsulin, you're going to get cleavage happening here and here, basically removing all of this section. Big chunks of it, like 70 amino acids or something like that, is lost there. And when that happens, you're just left with this little thin slice and this little thin slice. And of course, what's the only thing holding them together? These disulfide bonds connecting these cysteines together. So once again, the nature of the side groups of the amino acids are very important in determining the final structure of proteins. So like I said, we're not making you memorize the structure of all of these, but you should definitely know what the general classes are. And you should surely know what's special about cysteine, because cysteine does this actually a lot in proteins. Post-transcriptional processing is what this is called. Things that happen to proteins after they've been post-translational processing. So after the protein has been translated, it goes through additional processing steps to get it into its function. HIV, it will create one big protein. So it's kind of like a bacterium, and it has a lot of operons all stretched together. And in this case, it actually, instead of having stop codons in each of these places, it actually just makes it all as one big protein. At the end of the protein is a protease. The protease actually cleaves itself off the end of the protein and then proceeds down the rest of the protein to cleave these other proteins up, so that you get a reverse transcriptase and integrase and these GAG proteins. And so just another different way of processing proteins after the proteins have been made. In this case, the long sequence of genes includes in it the protein that's actually going to do the post-translational processing of the other proteins, which is kind of. Hemoglobin. You have alpha chains and beta chains. In hemoglobin, you have the heme groups. Each of these different beta chains and alpha chains, those are separate protein molecules, separate polypeptides. They get all folded up, and they get associated with one another into these big, what we refer to as quaternary structure. So remember from hopefully some chemistry way back, we have primary structure, we have secondary structure, we have tertiary structure, and we have quaternary structure. Quaternary structure is the structure when you're taking multiple protein chains and putting them together into more complex proteins. And so this is a representation of quaternary structure. Remember, primary structure is just the actual sequence of amino acids. Those amino acid sequences can form not very complex things like pleated sheets and beta helices and things like these. That is what we refer to as secondary structure. Tertiary structure is when that secondary structure begins to fold back on itself. So what you're really seeing, this is tertiary structure. Within each of these thick things is secondary structure that they're not illustrating for you. Pleated sheets, coils, things like this. And then these beta chains interacting with other beta chains and alpha chains, that's then quaternary structure. What holds together quaternary structure? I'm just going to tell you, this question is going to be on the test.
Non-covalent bonding.
Non-covalent bonding, these transient non-covalent bonds. What's holding together the tertiary structure? Brooks?
See above.
Yeah, see above, same thing. The secondary structure is often has to do with, in some cases, just the geometry of the amino acids that are being bonded together. In some cases, when you have helices and things like this, those helices are held together with non-covalent bonding. But simple pleated sheets, that's not due to any intermolecular attractive forces between amino acids. That's actually just due to the three-dimensional structure of the amino acids themselves. This structure breaks down really easily under heat. When you get sick, you get fever, what is your body doing? The body is trying to denature the proteins of whatever has made you sick before you denature your own proteins. When you're a baby and you get really sick and you start to get a fever of like 104, what do your parents do to you?
Threw me in ice bath.
They throw you in an ice bath, or after throwing you in an ice bath, they take you to the hospital because you're getting up into this range. I'm not saying that your parents were neglectful at all. That would suck being thrown into an ice bath when you're really, really hot. But you're getting up into this range where you're beginning to compromise these transient bonds that hold things together. These weak intermolecular forces. All right. Questions? All right, review time. Bacteria. Most control of bacteria is going to be occurring at the initiation of transcription. Because once again, there's one origin of transcription in bacteria, remember that? And then it's just genes all the way around. And so you tend to have transcriptional control. As soon as transcription starts happening, what happens relative to translation in bacteria?
It happens at the same time. It happens at the same time.
Yeah, as soon as you start transcribing messenger RNA, you start translating that messenger RNA into protein, because there's no transport of it out of the nucleus, because there's no nucleus to keep it hemmed in. And so most of your regulation of gene expression is going to be happening at the core promoter region, transcriptional control. Eukaryotes, not so simple. Regulation can occur due to things happening at the core promoter, where you have moderate levels of variation, can be happening at cis-regulatory modules that have a lot more variation from one cis-regulatory module to another. It can be controlled to a certain degree by modifications of histone molecules. So we have histone modification, we have that changes the structure of chromatin, we have transcription factors that bind to these cis-regulatory modules. That affects transcription. Once transcription occurs, other things that affect gene expression are the splicing patterns of messenger RNA after it's formed. Also, you can have these microRNAs. So you guys did talk about RNAi in lab yesterday. So you have these microRNAs that can either bond to RNA and just prevent translation from occurring, or when they bond to RNA, it basically signals that you should degrade that RNA, that the cell should degrade that RNA. If you don't get regulated that way, then there are also these translational mechanisms by which you have these elements in the untranslated regions that can suppress translation. So there are lots of different places where eukaryotic cells have the ability to put a stop on gene regulation. Or in some cases, do upregulation. But upregulation versus downregulation, there are a lot more opportunities to downregulate genes than there are typically to upregulate genes, because upregulation and downregulation both occur here and can also occur with chromatin modifications. But a lot of the rest of these things, like RNAi, that's suppression, translation-related regulation, that's all suppression for the most part. Translation in a nutshell. This is for protein coding genes. This is for non-protein coding genes, so non-coding RNAs. This would be transfer RNA, the different forms of ribosomal RNA. All of these things are still controlled by all of these things. It's just that because the end product is RNA, you don't have any of the regulation of translational regulation, because the end product is actually the RNA molecule rather than the protein. Questions? Anybody? Are any of you Taylor Swift fans? Excellent. Taylor Swift, I like her newer stuff more than I like her older stuff. I guess she was just interviewed by Elle magazine. I saw this in my news feed this morning, and I'm like, why are we interviewing Taylor Swift? Oh, because she's about to turn 30, like that's a news event. Evidently she's about to turn 30. So anyway, the topic of her of this interview is basically 30 things that I learned by the time I was 30. I think number 3 or 4 is that the cycle of failure and success, failure and success, failure and success is just a part of life. You fail, you try again. You fail, you try again. You fail, you try again. Somewhere in her life, she learned that that was a good thing to know. When you answer a question in class, because I'm peppering you with questions, take a little bit of the pressure off of yourself, because if you have the wrong answer, it's really just a teensy-weensy little fail. It lasts about a second and a half, and then we're moving on to the next thing. So I don't know that Taylor Swift has her life totally together. I'm not going to pass judgment on that, but on this one thing, she's got it pretty fine. Everybody makes mistakes. When we do these kinds of things where I'm really pushing you to answer questions and be the drivers of class, don't be afraid to give me the wrong answer. There's nothing wrong with that. My version of lesson 3, everybody fucks up something. Come ready to be interactive with one another and play with craft supplies, trying to get these things down. If you haven't reviewed the screencast from Monday that covered the first half of this chapter, you should definitely go and do that. Well, or was there too much flour in
More like, after he said like play. And I know I had a mistake in there.
I'll probably entertain a literature summary.
Okay.
Time to practice and make sure that it's
Cool.
Okay, you said you were gonna try to finish the.
Yeah, I think the diagram
That would be cool
of
because this.
translation, sure.
I mean, actually, when they know in person, is this like?
The current one?
Yes.
Yeah.
Okay.
And I'll do my best to help you with that. I myself. Sorry about that. It really is the paper.
I'm already.
I mean, if I
I've
don't see
already
my
committed.
Monday, that means that you guys.
So you found yourself.
I have long nails.
I think
I'm
You're really gonna have to be
I will not be
careful
sold.
this time.
They'll have to
Yeah.
show
So
me,
much
and
in
I'll
this
get some
paper.
evidence. I will not.
Katie, are
Yes.
you gonna come see me sometime?
I went to stop by your office yesterday, but you weren't there, so I.
Oh, what time?
2:30. 2:30, or it was 2:40. I was in that
Oh,
hall.
I have a independent study with 2 students from 2 to 3.
Gotcha.